Welcome, everyone. I know some people just jumped on again. We're Genetic ALS and FTD and the Legacy. We have the Professor Hautbergue here. We're so excited to have him and that he's taking some time at the end of a long UK workday, start of a West Coast America work day, to give us this little chat and opportunity to talk.
First of all, I'd say thank you, Jean, for inviting me to present our research, and thank you everyone for joining in. So maybe just a bit of background about myself. So I've been working on my PhD and postdoctoral research work on how genes are expressed in a normal condition, in a healthy condition. And working along that path, about 10 years ago when my institute, which is dedicated mainly on the work of ALS, has opened. So when my institute has opened, this is when I moved into setting up my group in CITRAN for Sheffield Institute for Translational Neuroscience. So basically the story I'm going to tell you about is I started around almost 10 years ago now. And I am an RNA biologist by training, and I will try to explain a little bit in late term RNA biology so that I can hopefully try to explain you the mechanism by which our gene therapy programs that we are currently developing are working. So on the next slide, please. So I'll just be starting by what is RNA biology, and this is a word that you will be hearing tonight. So what it allows, this process of RNA biology, it allows an essential process to all of the living units, which we call cells, whether this is in animal or in plant kingdom. And obviously the cells, which are those tiny little units in every single organism, they are allowed to produce the energy from the food we eat or the oxygen we breathe. And there are some sort of three important components for my talk today. So one is the DNA, which is a bit like, if you want, the house blueprint. It will contain all of the information which is needed for our cells to make what we call proteins, which are some of the building blocks of our cells, or which also make the machineries, which will make biological processes to happen and the energy to be produced. And in between the DNA and the proteins, we've got these molecules called RNA. And those are sort of little instruction manuals of strand of information, if you would like, which are copied from the DNA blueprint, and they have all of the information in order to make a protein. So on the next slide, so the way it's working in a cell, so this is in a normal functioning cells, we've got those two main compartments where you've got the nucleus in a gray. So this is the center of the cells, and this is where our DNA or the chromosome or this blueprint is contained within. And then in the surrounding space around the nucleus, there is this kind of jelly-like compartment, which is called the cytoplasm. And this is where all of the proteins are made. So the machinery that you can see here in red is what's called the ribosome, and this is what makes protein in the cell. So what you can see, therefore, is that for the RNA to make protein and be used within the protein synthesis factory, what you need is this RNA to be transported between the nucleus and the cytoplasm. And what's kind of important to give you a bit of an idea is we have about 21,500 protein coding genes in the nucleus of our cells, and this will allow to make up about 100 to 200,000 of proteins. So when we are trying to understand what's going wrong in a disease, it can really take a long and a lot of time to really pinpoint what is the defect and what you need to correct in order to develop a therapeutic approach. So on the next slide, I've taken an example of what happens if we have a change in the DNA blueprint, or you would probably know it's called a mutation as well. And this is what will cause genetic diseases and ALS in particular. So this is why you can say this little red cross, which I've highlighted on one of these chromosomes or the DNA in purple. What will happen is when the RNA is copied from the DNA blueprint, it will carry on it these changes or this mutation. And then when it will get transported into the cytoplasm, this change will be carried over into the protein which is made up. And what this will result is it will result in a faulty protein, which is not going to play its normal role into the cell. So that's how most of genetic diseases will be caused. And in ALS, we have over 25 different changes which can cause the disease. And in most of the cases, this will be this sort of producing a faulty protein, which is not able to work normally into the biological processes. And again, this single change will open in about 3.3 billion units of the blueprint. So you can imagine it's huge, and it's not really easy to pinpoint those changes. So now on the next slide, I have taken an example of C9orf72 ALS FTD. So this is 10% of all the population of ALS, and this is why indeed I completely agree with Dean. It's not a small population at all, especially if you take into account all the asymptomatic as well patients. And so in C9orf72, so you've got again this change, which is on purple in the chromosome, and this mutation or this change is a bit different, and we will see on next slide. It's constituted of long repeats, short repeats repeated many times. But what we found is that this C9 RNA is then associated with a protein which is called SRSF1. And this is a transporter protein which will allow the abnormal transport of this now C9 repeat RNA molecule into the cytoplasms. And there they will be producing some, again, abnormal proteins that we call dipeptide repeat proteins, because there are really small types of protein, so peptide, and they are sticky. So if you'd like, they maybe act a bit like a glue in a cell, and they will sort of freeze a lot of the mechanism or damage a lot of the biological processes. And those are extremely toxic and will end up by killing the cells. So on the next slide, I've tried to explain in a bit more what is the change in C9orf72 ALS FTD. So what you can see, it has these 6 bases of the DNA, so it's G, 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 C, C. So this is repeated up to 1,000 of times in patients. We can find it 2,000 to 3,000 or even 4,000 of those repeat expansion into the C9orf72 gene. And it's very, so the mechanism of action of pathogenesis is very complex. And there have been 3 different types of hypotheses which are non-exclusive. But clearly, all of the research so far, from multiple labs across the world, indicate that one of the key drivers of the motor nerve injury is the production of those dipeptide repeat proteins in a mechanism which is called RAN translation, because it's associated with the repeat. And so this is why I've made that, I've labeled this process in bold, because this is what we are going to be focusing on today and what my research has been focusing. So you can see that 6 different types of those DPR or sticky proteins are made from those, and those are the ones, the toxic components that will lead to the cell death. So then one of the aspects is it's very difficult to target the machinery that made the protein directly, because if you start off using drug against it, or it will also affect the production of other normal proteins in the cell. And this will usually have a lot of other bad effects on the cells. So what our strategy was is, if we understand how the C9 repeat RNA gets into the cytoplasm, then maybe we would be able to target this mechanism. And if we can prevent those repeat RNA molecules from going into the cytoplasm, then the cell won't be able to use them to produce a toxic protein. And so this is how we build up our research. So on the next slide, what we hypothesize indeed is that excessive stickiness of that SRSF1 protein onto the repeat RNA will allow them to be transported into the cytoplasm and be used by the machinery that produce the protein to make those toxic DPR. So once we've had identified that SRSF1 was one of the key proteins by binding those transcripts, I'm going to try to show you just a few experiments showing that, yes, this protein is involved into this process, but also, and more importantly, in terms of a therapeutic approach, can we try to change the way SRSF1 is working so that we can potentially use this to develop a novel therapeutic approach. And so this is what we embarked on about, so it was 2014 when we started on that work. And on the next slide, I'm going to show you one of the first experiments. So what we started is we used just some neuronal cell models, which are quite easy to grow in a dish, in a lab. So you can grow them just in a few days, so they are quite useful when you start doing experiments, and you can also produce millions and millions of those cells. So it means you can do biochemical type of experiments, which usually require quite a lot of materials. So those cells, what we can do is we can just introduce. So here we've had either 38 G-force into repeat, or what I didn't say yet is that we can also have the antisense repeats, which are also made. So this is why those are called C4G2. And we've had 39 repeats of those. And all of those black sort of bonds you can see in the first two top panel, GP and GA of the experiment on the left, they are some of the proteins of those sticky dipeptide repeat proteins that we can see in our experiments. Now, what you can see, what is called SRSF1 RNAi, or it means this is when we're trying to lower the amount of the protein SRSF1. So each time you see a little plus, you can see that there is less of those sticky proteins. And so the principle here was that if we have excessive stickiness of that protein onto the C9 repeat RNAs in pink there, so if we have too much of that, if we just reduce the amount that we have in a cell, then it should reduce the transport into the cytoplasm and therefore reduce the production of those toxic protein. And this is exactly what we were able to see into this cell model. Now we can use those cells is when we, so if you look at blue bars, they are just normal cells. And then when we introduce those repeats into the cells, you can see on the green bar or red bar, the first bars that they are lower because cells are not growing as much. Now where we are able to, when we have the SRSF1 lowering experiment, you can see that those bars goes up because they make the cell grow faster again. So this was just a sort of very quick and easy experiment to kind of see that, yes, decreasing the total amount of SRSF1 ends up in decreasing the total amount of the toxic DPRs. And so then on the next slide, we moved up into using a more complex system. So what we can do is we can take skin biopsies, and our research wouldn't be possible without the patient, giving us skin biopsy. So we are always very, very grateful to patients taking part in our research, because then from those skin biopsy, we are able to then reprogram the skin cells into either astrocyte cells or motor nerve cells, so cells which are into our brains. And we can do that in a dish in the lab. So those are fantastic models because they contain the blueprint, the exact blueprint of the patient, and we can really study nerve cells in a dish in the lab. So this is what you can see here. We have mixed together some astrocyte in red from the ALS patient, together from motor nerve in green from ALS patient. And in this system, what we see, as you can see in a control cell versus familial ALS, FALS, or sporadic ALS for SALS, what we can see is that in the ALS context, the motor nerve cells are dying by about 50%. So this is a kind of a very good system for us to test our therapeutic approaches, because if something is working, it should stimulate the survival or allow the survival of the ALS nerve cells. So on the next slide, this is what we tested with lowering or reducing the amount of the protein SRSF1 into motor nerve cells. So what we've got is we have a situation, so the top left corner is our healthy individual situation. So we've got, again, astrocyte in red. We've got the nerve cells, which are in green, and you can see the arrow points to an axon, which is those processes that nerve cells are doing where the electric signal is conducting from one to another cells. What you can see is that when we then move to the top right panel, so this is in an ALS condition, you can see that we have less green cells because they are dying in these conditions. And this is in the C9 ALS context. Now, if we deplete or if we reduce the total level of the SRSF1 protein, so this is what's called delta SRSF1 in the bottom panels, then you can see that now we have a lot more green cells in the ALS condition. So this is this is the bottom right panel. So this is showing that lowering or reducing the amount of the SRSF1 protein in the motor nerve cells allows to stimulate their survival. And the red, orange, and yellow bars are actually showing 3 different lines, which were taken from 3 different patients. And you can see that in each of the cases, we have more motor neurons surviving in this condition. And if you look at the sticky DPR at the bottom, what you can see is that we have less of the black signal when we have used our treatment to reduce the SRSF1 protein. So this was really encouraging because it's indicating that not only this is working in our easier cell model, but it's also now working into the genetic makeup of a patient with C9 ALS. So then on the next slide, we looked at, does it really change the transport of this toxic, repeat RNA? So what we have is we have in red some healthy nerve cells, so generated from healthy individuals. And T, N, and C represent total, so that's what you see in total in a cell if you try to measure that RNA. N is what we measure in the nucleus, and C is what we measure in the cytoplasm. And what you can see is that in a normal condition, there is obviously no repeat RNA, so there is nothing going into the cytoplasm. Now, when we move into the blue condition, so this is a C9 ALS condition, what you can see is that the first blue bar is really big on C, on the cytoplasm, because a lot of those C9 or 72 repeat RNA molecules have been transported into the cytoplasm. And when we reduce the amount of the SRSF1 protein, so this is what's called in the condition LV SRSF1 RNAi, so where we have 5, so we add 5 virus for one cell, which allows to lower the SRSF1 protein, then you can see that the big blue bar in C becomes very small into the other C condition, while on the opposite, in the N, it becomes bigger. So it's indicated that we have less amount into the cytoplasm, more into the nucleus, because we have now stopped its transport. And this is exactly what we wanted to check, that we understood properly the mechanism by which lowering the SRSF1 protein is working. So having some very nice results in cells, we then decided to move into fruit fly models on the next slide. Oh, no, sorry. So what we did before that is we just checked. So you could imagine that reducing the level of SRSF1 may have some other bad impact on other RNA in the cell. Because I've said before, so we have over 200,000 RNA molecules in a cell. So what we wanted is to check this. So we have some techniques which are called next generation RNA sequencing or RNA-seq. I don't know if you've heard about those, but we are able to know exactly all of the RNAs that are present in a cell and what are their relative amounts. So what we did is we took neuron or nerve cells which were either treated or untreated for the reduced amount of SRSF1, and then we've looked at all of the RNA in the cell. And actually what was really nice is, so as you can see in a C9 nerve cells, 2,268 RNAs were changed in the disease, so between normal, healthy, and just disease. And when we reduced the SRSF1 protein, then we could see some other changes. And what was really nice, so if you look onto the left of the right panel, is you've got in red that's all of the RNAs which were affected in a bad way in the disease, so they are reduced level, while in green they are all the ones which were going up, so too much of them. Now, when we treated our C9 nerve cells with this virus, which allowed to reduce the SRSF1 protein, you can see that what was red becomes green, and what was green becomes red, because we are actually restoring the normal level. So this was a very nice result that gave us confidence that, first of all, there were only few changes, so we were not changing completely the whole gene profile of a cell, but that also what we were doing was exactly what we wanted, so it's reverse the bad things into the good things. So this is when then we decided to move on to some animal models experiment. So on the next slide, we started with, oh, no, sorry, I did the animal later on. So this was, the first strategy was about reducing the amount of the SRSF1 protein. Now we have been thinking, because we just want to see if we can have several approaches on the same target. So rather than changing the level of the SRSF1 protein in the cell, we thought about not changing those, but use what is called a cell-permeable peptide. So it's a very little protein which will stop the SRSF1 protein to contact the machinery, the transport machinery. So if you'd like, it's a little bit like a drug-like molecule, which will stick to the transport machinery and stop SRSF1 for contacting it. And so if we do that, we would obviously, we were expecting that SRSF1, which was stuck onto those C9 repeat RNA, is not going to bind or to contact the transport machinery, therefore won't get into the cytoplasm, and it's not going to make the neurotoxic protein. So this is what we did there. We use this by just adding this drug-like molecule into the medium of our cell model that you saw before. So that was the first cell model. And what you can see is that the bonds into the right panel just goes down as we increase the amount of cell permeable peptide, or CPP, that we add into the medium. And this was very nice because it fits to a very nice dose-dependent reduction, as you can see on the graph below the bounds. And again, as I've shown before, so we have bigger green bars and red bars, so it means that our cells grow better when we add this peptide, this CPP peptide, into the medium of the cell. So this was a second strategy to tell us that we don't need to change the level of SRSF1 in a cell. We can just block how it contacts the transport machinery, and it gives exactly the same result. So that gives us a lot of confidence that SRSF1 is indeed the real protein we want to target in a therapeutic approach. Because as I said, we have 100 to 200,000 proteins, so you need to make sure what you observe is not indirect. So this was the proof of the direct involvement of that protein. But also it gives us two therapeutic approach potentially to go on into stopping those repeat RNA from going into the cytoplasm. So then on the next slide, this is when we started our work into animal models. So we started by using fruit fly. So we can introduce the same changes, so 36 repeat changes into the genome of fruit fly, and what you see there are the eyes of the fruit flies. Yeah. So wild type are the same as healthy flies, so you can see that they have a very, very nice eyes with their ommatidia, or their little eyes, nicely aligned. Now, if we take what's called G4C2 times 36, so this is the ALS model. And as you can see, this one, the flies develop those black spots onto their eyes. And it's because the nerve cells of the eyes are dying. So for us, it's very convenient in the lab because we just look at the eyes and we can do a lot of genetic in the fruit fly model. And then in order to test our therapeutic approach. So what you can see on the extreme right is that this time we are reducing the amount of SRSF1 in the eyes of the fly, and you can see that the eye becomes just completely normal when we reduce the SRSF1 protein. So this was, again, really great result because it shows that you can stop the neuronal cells, the nerve cells, from dying in vivo in an animal, even if, of course, fruit fly is very different from us humans, but it was the first good start of the experiment, because if it doesn't work at this stage, there is not even any point in going further. Yeah, it's not going to have any chance of working in more complex organisms. So then what we, so that stops the nerve cells from dying. And then on the next slide, so the flies are quite convenient because what you can do is what we call climbing assays or climbing experiments. So you can put the flies in a tube, and then if you tap the tube, they will all fall at the bottom of the tube. And then you can just time up how long it takes them to climb the tube. And obviously, I didn't need to present here what is ALS or FTD to this audience, but this is kind of mimicking the locomotor deficit that would be observed in a patient, because those flies are not able to climb as fast as normal. And this is what you can see in the red bar, we have our C9 ALS model flies, and they climb slower than what's called the WT or the wild type, the healthy flies. Now, when we deplete, when we reduce the amount of the SRSF1 protein in our flies, what you can see on the right bar, which is in khaki, so you can see that now those flies, they climb as fast as the normal flies when we have reduced the level of the SRSF1 protein. So this was really telling us that not only it stops the nerve cells from dying, but it's also restoring the motor function in these animals. So those were really exciting times when we could see this data. So then we were encouraging into moving further into mice. And on the next slide, so this is where, before we did that, there are some images not showing, but it doesn't matter too much. The bar charts are more important. So before that, we wanted to also check that our other strategy, the CPP, so the drug-like peptide, was also working in the Drosophila and in the nerve cells. So on the left, you can see that if we add this drug-like or CPP into the food of the Drosophila, then the red bar shows the flies which are not climbing as fast because they are the C9 ALS model. And then again, in the khaki bar, you can see that when we feed the flies with this peptide, this CPP molecule, then they climb again as fast as the healthy flies, really showing that both therapeutic approaches, reducing the level of the SRSF1 protein, or stopping it from interacting or binding the transport machinery, both of those options would work. And we've done the same experiment with the motor nerves culture, patient-derived motor nerve culture, as I've shown you before. And the light green bar was the result when we used reducing the amount of the SRSF1 protein. And if you see the dark green bar, which is treatment with the CPP, it gives exactly the same result. So again, highlighting that we have those two therapeutic approaches that we are now following up in the lab in parallel. And this was the result which were presented in the latest of the publication in Science Translational Medicine, which all of the reduction of the SRSF1 protein was presented a few years ago in also a very prestigious journal in Nature Communication. And it's really because we've got those two approaches, head to head. And we will now be testing if one is based on the other, if both work, can we do a combination, or we don't know. That's the kind of things we will be following up. But then on the next slide, what we have started to do is, so we've got great result in, in preclinical models such as the patient cells, nerve cells, in the fruit fly. So we wanted to kind of now move to a more complex organism, which will be mice. So those mice that we are using, they have a human C9orf72 ALS gene into them. So they were built by Laura Ranum in the University of Florida, which is a collaborator of mine, and we have her mice. And so those mice that produce all of those sticky dipeptide repeat proteins. So they are obviously a good model for us to see whether lowering or reducing the amount of SRSF1 or whether injecting these peptides would work into a more complex organism. So the way we do gene therapy experiment in mice, so it's much more closer to what it would be if one day we reach a clinical trial. So we are using on SCAAV9, so this is on adeno-associated virus 9. So this is a non-pathogenic virus for humans. And what we do, we engineer it. So we remove the bits of DNA in the virus that we don't need, and we actually replace them with a bit which will allow to reduce the amount of the SRSF1 protein. And then we use this, we produce this engineering engineered virus, which is here in black, and we can inject it into the cerebrospinal fluid, or CSF of the mice. So via a position which is called, where you see the syringe here at the back of the brain, called cisterna magna. So that would be just a bit behind our neck for us in human. In human, you probably know clinical trials with antisense oligonucleotide, for example, are done via lumbar punctures for a lot of them, because lumbar puncture is safer in human. But the principle is the same. The therapeutic agent is delivered into the cerebrospinal fluid. And that will allow all of the brain and the spinal cord to basically be in contact and bathed into your therapeutic agent. So in our case, our AAV9 virus, which allows to lower the amount of the SRSF1 protein. And what you can see on the bar chart, then we've injected some mice, and we can check where is the virus going, and we can see that we have a lot of virus in the brain. We have a bit less in the spinal cord, which is kind of expected because it's a bit further away from the site of injection. And actually, this is something that still requires a lot of research. So that's what we call the route of administration, and how much virus goes into the brain, into the spinal cord. It's still a very, very active of research. And because gene therapies are really so promising these days, and start to generate transformative results, for example, in spinal muscular atrophy, which is a juvenile or a childhood form of motor neuron disease, that I expect those to explode over the next few years, and probably have new devices, medical devices, and so on, be developed to actually help clinicians deliver much more efficiently and safely therapeutic agent into the spinal cord. But so this is great because we can see that we've got nice enrichment, nice distribution of our virus in the brain. And there is a little bit in the heart or muscle, and it's known that the virus can spread to some other organ. And this is obviously things that in our study, we are not only looking at the brain and spinal cord, but we will be looking at all of the organ in mice in order to be sure that our agents are not damaging other organs. Just switch on my light again, sorry. So on the next slide, we did an experiment there where we've injected our virus into the CSF. And those are just, we can then, so 2 months later, we kind of take the brain. So here what you can see is a part which is called the cerebellum. You can see in blue, that's a staining of all the nucleus. In green, so we can stain our virus in green. So you can see we have lots of virus, which is obviously exactly what we wanted. We wanted our virus to have gone from the CSF into the brain, into the nerve cells. You can actually make details of some nerve cells, which shows nicely, and then it's a merge of the 2 images. And if we looked at, did it work? Did we reduce the dipeptide repeat protein level? So what you can see, what's called NTG, it's a non-transgenic mouse. So it means those are mice which don't have the disease, don't have the change. So obviously, they don't produce any of those toxic sticky proteins. In red, this is just a C9 ALS mouse model. So you can see it produces a lot of those sticky dipeptide repeat protein, which we normalize to 100% there. And then in orange, this is a control virus, which doesn't lower the SRSF1 protein, doesn't reduce SRSF1. So as you can see, it has no effect onto the DPR as expected. But then we have injected 2 different doses in green of our virus, which does reduce the protein SRSF1. And what you can see is that it also nicely reduces the toxic DPR into a dose-dependent manner. So this is obviously very exciting because it's showing that this is also now in a mouse, and we've not detected bad deleterious effect up to 6 months at the moment. We've conducted the longest study. So what we have also done is injected on the next slide is injected the peptide directly into the CSF just to test whether it would also lead to the reduction of the dipeptide repeat protein of those toxic proteins. And so what you can see is that, so we've got the same, we've got the nucleus in blue, untreated on the top panel, and then with the peptide on the bottom panels. And so we can see the CPP in green. So you can see that again, it has gone within the cell inside the brain where we wanted it to go. And then when we look at the bar chart, which looks to have lost its color, but doesn't matter. So we've got again the non-transgenic, which have none of those sticky proteins. Then the C9 transgenic, which used to be the red bar before, does express a lot of those DPR. The control peptide doesn't do any effect. So this is another type of peptide which doesn't reduce those sticky proteins. But when we inject the SRSF1 peptide, which is the last bar on the right, then you can see that now we have a nice reduction of the sticky proteins. So basically, to conclude, I think I'm just in the last, on the next slide. So we are really excited that we have two therapeutic approaches we think that we can take forward as gene therapy approaches. One is kind of based on a viral strategy. So this means you would have a one-dose injection, but it's obviously more invasive because you need to do a lumbar puncture or a cisterna magna injection. But that's for, let's say, life. You just do it one time. But we also have an alternative approach, which could be to use this cell-permeable peptide or CPP. So this one wouldn't be that invasive because you could potentially use as a nasal spray. There is a lot of research done at the moment into this, but you would need to use repeat dosing. So we kind of have a sort of nice portfolio and balance. So we kind of trying now to follow up these into longer study, larger number of animals, so that we can test head-to-head all of those therapeutic approaches. And this is a work that I've been doing in collaboration with Prof Mimoun Azouz and Prof Pam Shaw, who are also working in Citran in the same institute as I am. And so what are the immediate next steps on the next slide? So we are completing a larger safety and efficacy study in mice, as I was telling you. So now we are going to do some 12-month study on a very large, I had about 6 months in the experiment I've shown you. We will now have 18 mice from each group. So in order to check that the animals are not sick, that it still reduces nicely the sticky protein 12 months after the injection. And this is currently a study which will run until 2024 and is currently funded by LifeArc and Motor Neuron Disease Association. In parallel, we have already starting to meet with some regulatory bodies. So it's like the MHRA in the UK, but we're planning to approach the FDA in the US or the EMA in Europe, so that we can present our data and make sure that we will generate within the next 2 to 3 years data that could potentially allow us to run a clinical trial if the outcomes are what we expect. So now to conduct a clinical trial will require a first-in-human trial, about 15 to 20 million pounds, which would be around the same, yes, in US dollars. So this is obviously a very large sum, and it's very difficult for academics to find this large sum of money. So I'm actually the founding director now of a university spin-out company which is called Crucible Therapeutics Limited. So it was incorporated in February this year. And this is done with again, Prof Mimounazous and Prof Pamcho, who are the other co-founders. And so what we hope with that is use the spin-out company as a vehicle in order to carry on the more expensive research that would, for example, be needed into non-human primates to test that our product is ultimately safe. But also a mouse is very different from a human. So we will need, a model in between which is bigger, which will allow us to test our dose so that we know what dose we would be using if it's working all perfectly well in mice, and what dose we need to use into a patient. So we are just in the process of closing a deal for a 5 million seed of investment. That this would allow us to then manufacture our vectors in huge, in very big amount, which would allow us to do the non-human primate study. So these types of studies, just 2.5 million, just one study, and then apply to our for clinical trial and perform the first trial, hopefully in 2026, 2027, if everything goes as expected, and if we found that it is safe, but also, continues to be efficacious as it has been so far. And then with that, I just like on the last slide. So this is just to acknowledge, multiple people who are involved into this study. So I've got, postdoctoral researchers, Dr. Lydia Castelli, Dr. Yahweh Lin, Wan Ping Wang, Kamalia Imram, and Dave Burrow. They are all instrumental and have all been working, whether in the cell models or in the animal models, into this project. Also, talented PhD students, Charlotte Mason, Bridget Benson, Eitach Gul, Nikita Soni, and Sajid Shah. We got help from fantastic technicians. Chloé Moutin, Ergita Bali have been all working onto the SRSF1-based program, as well as some undergraduate technicians with Chris Dimeki and Abby Famin. So I'd like to thank my collaborators, so Prof Mimounazous and Prof. Then Pamela Schwartz, I've already said, but as well I have done a lot of work with Laura Ferreiro and Richard Mead and many other colleagues in the Institute. To acknowledge my structural biology, fly research, and all of the fundings from those different academic and more translational funds, which in which the research wouldn't be possible without that. And obviously, as I already said in my talk, I'd like to really acknowledge all the patients who give us biosamples, because our research wouldn't be possible as well without them. And thank you all for your attention.
Wow, thank you so much, Professor, and we're all thankful for your team as well. So please extend our thanks to all of your collaborators and team for all the work you're doing. We have a few people with questions. I want to take, I have a bunch, but I'll take the privilege as the hostess for just one to start, and then I'll go to our audience. Do you think that the CRF1 is also somehow implicated in TDP43 mislocalization into the cytoplasm? I'm not really a scientist at all. Yes, I don't really understand these things.
This is an excellent question, and this is a very difficult one to reply to. We don't know is the simple answers. We are looking into it. The problem is that we don't really understand very well the correlation between TDP43 proteinopathy and the DPR. So it's obviously very difficult to then assess the therapeutic effect on SRSF1 directly onto the TDP43 proteinopathy before we know a bit more of the relation between the DPR and the TDP43 proteinopathy. But what's really linked to that, and we only have some thought, is that we think that potentially SRSF1 could be used more broadly, not only in C9 potentially. So this sort of big genome-wide studies that have been presenting, where we could see change reverted, actually highlighted that just reducing the amount of the SRSF1 protein is kind of good for neuronal cells and nerve cells. So this is something that we're going to investigate into trying to extend to potentially other type of ALS, but also why not to other neurodegenerative diseases. And so we have a few programs starting, but they are way behind compared to our work on C9 ALS FTD.
Wonderful. Thank you so much. We are approaching the hour. Are you okay to go a little over?
Yeah, I'm fine. I'm fine. Absolutely fine.
So Paul, do you want to go ahead? And if anyone has a question after that, I think if you could see the raised hand, that would probably help keep things in order.
Sure. Can everybody hear me okay?
Yes.
Great. First of all, thank you for making me regret not paying more attention in biology class. Clearly, that went, a lot of that went over my head. At the very end, you talked about the clinical trials. One, are you collaborating with any U.S. universities that might make it easier to get some U.S. funding for this? Number two, sign me up. I'm ready to go. I got little my mouse ears. Okay. Just pretend I'm a mouse. Okay. Is it possible, seriously, is it possible to start lining people up before you complete the second mouse trial?
Of course, yeah. So the first thing is obviously any trial will be dependent on data in the monkeys as well as in the mice. So this is why we wouldn't get those results before 2 to 3 years. So the first question was about if we collaborate with other universities, and so the plan it is at the moment is the first, because we do a lot of clinical trials in Sheffield. Professor Shaw is one of our lead, is a lead clinician scientist, and has been involved in something like over 20 clinical trials in ALS. So the plan would be in 26, 27, if everything goes accordingly, to do the first clinical trial in Sheffield. We also have a new facility which is led by Prof Guillaume Hautbergue, where we can manufacture the GMP grade vectors that can be used for clinical trials. So we kind of have all of the basic research with the manufacturing facility and the clinical trial center in Sheffield. So this is a plan for the first trial, which would only involve a few patients at first. So we think it would be 12 or 15 patients. So most likely more local UK patients. But then obviously after that, we would need to go on to a phase 2 trial. And then the plan is indeed then to go to US, collaborate with a lot of other persons, and I know quite a few people through the US, but also open it to Europe, yes, not only to stay in UK, where then we could test in many more patients. But obviously all of this will depend first on the mouse and monkey result, and then on the first of the results on the first trial.
I can travel to Sheffield. It's very nice there. Thank you very much for your work on this, and good luck, and hopefully Gene will pass along whatever information you can provide on this, and we're rooting for you.